Welcome back, students. This is Module 2.4.5, Types of Photosynthesis. We're going to describe for you three types of photosynthesis, starting with what is called C3 photosynthesis, or what you can think of as regular photosynthesis. It is so named because the first product of the dark reaction is a 3-carbon compound. And this is contrasted with at least one other type of photosynthesis that has a differing number of carbons for the first product of the dark reactions. At room temperature, C3 plants lose about a fourth of their fixed carbon to photorespiration. In other words, their efficiency in making glucose is reduced by 1/4. Wow, and that's all from photorespiration. So that's one challenge, one problem that has to be addressed. And the thing is, at warmer temperatures, it gets even worse. At higher temperatures, photorespiration increases faster than photosynthesis, and C3 plants can lose up to half of their photosynthate, or the glucose that they would make with the process. Now, you must remember that photorespiration is a competing reaction that has no useful purpose in plants. It is only an inefficiency. And to drive this home, think of your paycheck. Suppose you're making $400, and instead you get $200. That's what we're talking about. It is a significant reduction, a significant inefficiency. And so because of that, other pathways of photosynthesis have evolved, and so we can see that there are these other mechanisms, and they're beyond C3, or regular photosynthesis that we just described. There's C4 photosynthesis and CAM photosynthesis. We told you one problem is photorespiration, but what makes that even worse is what happens to plants in a hot environment. So under hot, arid conditions, these will lose water by evaporation through these leaf pores that are called stoma, or they can be called a stomate, and plural stomata. And so if you open these, and why would a plant need to open its leaf pores? Well, if it's photosynthesizing, it needs carbon dioxide. And as they open the leaf pores, the stomata, water vapor is lost. This can be very serious, where primary productivity is limited by water. And so if the stomata close to conserve water, oxygen will build up inside the leaf, and carbon dioxide can't enter. And now if you get a higher concentration of oxygen, remember that photorespiration is a competition between oxygen and carbon dioxide for the same enzyme. And when oxygen is higher concentrations and wins the competition, then photorespiration results instead of the dark reactions of photosynthesis that makes glucose. And so this is really compounding the problem of photorespiration. And so this actually is an illustration which shows two mechanisms, C4 photosynthesis on the left and CAM photosynthesis on the right. And C4 is really a spatial separation, while CAM is a temporal separation of the steps of the process. So let's look at C4 first, and we'll go back to that figure. C4 is a spatial method to favor carbon fixation or photosynthesis over photorespiration. It's called C4 because the first product in the dark reactions is a 4-carbon compound as opposed to a 3-carbon compound. C4 photosynthesis is used by many grasses and also some other plants, such as members of the yucca and agave families. And I said it's a spatial, that means in space, and what C4 primarily does is it concentrates high local levels of carbon dioxide to favor carbon fixation over photorespiration. And it does this by transporting carbon dioxide to special cells that are found around the veins inside the leaves of C4 plants. These are called sheath bundle cells, or bundle sheath cells. And they actually are airtight, and so when we transport carbon dioxide in, the concentration is very high in there. There is a cost to this transport because it is active transport, but even so, it is much more efficient. So carbon dioxide concentrations increase in those bundle sheath cells, and it favors photosynthesis over photorespiration. And there we're talking what I was saying earlier about the cost. But even so, it greatly increases, that is C4 photosynthesis, greatly increases the efficiency of photosynthesis in hot climates. And that's where C4 plants really have an advantage. We can compare now the spatial method, I'm sorry, that was a spatial method, the temporal method of CAM photosynthesis. CAM, by the way, is really this alphabet soup name. It stands for crassulacean acid metabolism. And I'll show you a picture of the plants in the family Crassulaceae, which is what this is named for. It's where this process was discovered. And as I said, this is a temporal method to favor carbon fixation over photorespiration. And remember, carbon fixation is photosynthesis in the dark reactions. It's used by cacti, agaves. I misspoke when I said agaves were on the previous. That was a mistake I made, so make sure you don't put that. But agaves are found there. Bromeliads, including the pineapple, and other members of that family. Many drought-adapted plants do use this method. The challenge here includes water loss as well as photorespiration. When plant opens its stomata, gas exchange occurs, which is good, but so does water evaporation. Bad, as that previous slide showed. And so there's always a tension in plant physiology between gas exchange and water loss. Now, how do you solve that? Well, the CAM plants do it temporally. They open their stomata at night when water loss is greatly reduced, the temperatures are lower, the relative humidity is higher, and they bring carbon dioxide in and they store it as an organic acid. And by continually converting carbon dioxide to this organic acid, it allows carbon dioxide to continue to fuse into the stomates. So that's very interesting. And so it reduces photorespiration, it increases the concentration of carbon dioxide, because during the daytime when the sun is shining, the plant will convert those organic acids back into carbon dioxide, and will have a high concentration of carbon dioxide inside the leaf without the water loss that is often associated with that process. And so here I told you we'd go back to this slide. Spatial separations. So where it says Calvin cycle here, that's another name for the dark reactions of photosynthesis, and that's where the sugar's coming out. So think of this as the dark reactions. Here it's separated in space. A typical leaf cell is called a mesophyll cell, and we have to transport the carbon dioxide to the bundle sheath cell, separated in space. Here, in CAM plants, the process is separated by time. The carbon dioxide is concentrated at night, and then in the daytime, the stomata, the leaf pores, are able to remain closed, and the products of the light reactions are then able to drive the dark reactions. Here, by the way, I told you I'd show you a picture of the Crassulaceae. This is called the stonecrop family, and that's where CAM photosynthesis was first discovered. And that ends this module.